Hi, we're going to talk about sample collection methods and especially sample collection which are to be used in the applications of forensic sciences, for clinical SNP genotyping or for security measures. The main purpose of sample collection, biological sample collection, either it be blood, semen, sweat or any of the biological fluid, is to generate a DNA profile to identify the identity of the suspect. There are quite a few different type of sample collection devices. The most common sample collection device which are used is based on the common Q-tip. The common Q-tip, and there are different versions of the Q-tips with a longer handle, but the most common version of it is the cotton swab at this end. And the various versions of the cotton Q-tip swabs are the nylon swabs and rayon swabs and cotton flock swabs, and multiple swabs and dispensers which can be used with a double applicator swabs also. The purpose of this demonstration today is to show and demonstrate a different type of swab collection device which is totally different from the swab sample collection device. It is based on a matrix which is liquid in nature, and we will show you a little bit later about the use of the Omni-Matrix as a sample collection device. In sample collection device, so in addition to the biological sample, which we just talked about, the different types of biological sample, there are different type of substrates where the biological sample is actually deposited. The most common in a forensic environment is on clothing or in any of the solid surfaces. The clothing, if a biological sample, and in this case we have deposited some of it's 2 microliter of diluted blood. This is in a cloth which is deposited. Cloth gives an example of a porous surface, and then we have a metal surface. In the metal surface, and we have deposited 2 microliter of diluted blood. And if you notice that I'm holding this knife, during the process of just holding it, I'm depositing cells which are being shed from my skin or hand and being deposited here, and then and I'm intentionally pressing it here, and then we will show you. Similarly, we have deposited blood samples just to make it visible. It could be invisible saliva or semen or any of the other biological fluid on a wood surface and on a plastic or glass surface. So these are the different type of substrates. From a porous substrate, usually the practice is to actually cut it out, the sample, where we can visually see it. Whereas from any of the solid surface, a swab is used. And we will show you the use of the swab in session 2, where actually the sample is being collected for processing. Thank you. Hi, this is session 2 of the sample collection devices. In this session, we are going to actually introduce a new sample collection device which we call as the Omni-Matrix. And in the previous session, we showed you the sample collection devices which are all based on a swab. These are the different swabs, and the nylon swab or rayon swabs and so on. And I will just show you one which is the most common one, is the forensic swab, which is flocked at the end over here, and we are going to open it later during the actual use. So these are the different swabs, and actually I'm just going to show you in a way is the sample. If this is the sample where it has to be collected, so we will take a swab and have this mosh in the sample area and then just swab it. In session 3, we will actually show the sample collection method. So this is the classical method of using swabs, any of the different swabs in collection of the sample deposited. If it is like in the previous session, I showed you about the fingerprint, if there are fingerprints on any of the non-porous surface, similarly a swab is used to collect the sample shed. Whereas we are going to introduce today a new type of sample collection, what we call as the Omni-Matrix. The Omni-Matrix is a liquid fluid, and it is supplied as a kit. It is manufactured by Biofunctions, and in the kit, you will see, and we have it already outside, here is a dropper bottle with the Omni-Matrix inside, and the concept is you take if the sample type is visible, and you just go and apply as a drop on the sample. The other type that Omni-Matrix is supplied in the kit is a small spray bottle. If you notice, these are the small dispenser type bottles and the spray bottle. Each one of this is for only one session. So if there's a sample collection session now, we will use one of these bottles and then we can discard it. And a spray, when an area is barely visible, like a fingerprint, so we can just go and spray those areas and then we actually scrape it off from the surface. And we are going to show you how to scrape it. If this is an area which is already sprayed and there was a fingerprint here, we will pick it up like this and then deposit. And in the next session, we will show you actual demonstration of sample collection, and then we will proceed into sample processing also. The main feature of the Omni-Matrix as opposed to the swab is in the application with trace amount of DNA sample or the trace amount of biological fluid. What happens is in the sample collection, and when we are talking about trace amount, we are talking about usually fingerprint, or there are hardly any even fluid, biological fluid. It has been documented about 30 to 80% of the sample is actually lost in the collection procedure, and then another 20% is lost during the processing. And we are going to show you a very efficient process using the Omni-Matrix, where about almost 90% of 90 to 100% of the sample is collected, thereby generating. We also have data to show from a fingerprint and multiple fingerprints, which is probably around 20 to 50 cells, where we were able to generate a reliable DNA profile with almost 100% alleles showing there. Thank you so much. This is session 3 of the sample collection devices. In this session, we are going to concentrate on the actual collection of the sample. And we are going to show first the classical method, which comprises using the swabs. This particular swab which we have selected is manufactured by Puritan, but there are other swabs also by Board Technologies and Forensics swabs and quite a few other swabs. The first thing with the swab is what we do is, and these are the different samples which have to be collected. This is in a plastic surface which we have already have the samples dried out. So the first thing what they do is in sample collection is actually moisten the sample, and if it is dried out. In moistening the sample, usually it is just simple water or TE. So in this process we have moistened the samples, and then we're going to take out a classical swab, and then we are going to swab the surface. Usually if the sample is in trace amount, you won't be able to see any leftovers. If there is any leftovers, I'm just going to concentrate here. Most of the sample is collected on the swab itself, and the leftover, there's hardly any left there. But usually about 20 to 30% or 15 to 20% of the sample is actually left there because it was unable to swab. After the sample collection, it is allowed to dry on a benchtop or in the supplied container. This is the supplied container, and this is transported. Now we are going to compare with the new Omni-Matrix sample collection system. In the Omni-Matrix sample collection system, what we have done is actually after the sample collected on the swab has totally dried, this swab is inserted into the container and transported to the lab, where it's going to be processed for DNA extraction. The Omni-Matrix is supplied, as previously in the session, in the previous session, in 2 tubes. One is a dropper bottle, where it is in the dropper bottle is that like this, and the other one, it is in a spray bottle. In the spray bottle, it is meant for spraying. What we have done in the meantime, prior to this session, we have already deposited a blood sample which was used for the swab also. We're now going to use the dropper, and with the dropper we're just going to drop 1 or 2 drops of the Omni-Matrix, sufficient to cover the the sample area, and we are going to cover all the samples which are collected, which are in the surface of the area. Similarly, we will do it on the wood surface and on the metal surface. Show you trace DNA collection where you actually the DNA sample or the biological sample is not visible. For instance, if we suspect that this area or the handle area of the knife, there must be DNA sample, so we will take the spray and spray it on that suspected area. Similarly, on the wood or any other surface where we suspect that there will be biological sample, we spray on those areas. These matrix, the Omni-Matrix, either the spray or the one which we deposited earlier on the non-porous surfaces, they take about 3 to 5 minutes to dry out. After they are dried out, depending on the humidity of the surface, so then they are actually scraped off. Now we are going to show you actual collection of the sample with the Omni-Matrix, which was in this case applied with a dropper. So we can just pick it up, and after picking it up, it will be just deposited directly. And if you notice that almost 100% of the sample is picked up, in this case it just flew over, which is okay, and it is deposited into a microtube. Actually, these are the samples which were previously applied with the Omni-Matrix, and we let it dry for 3 to 5 minutes. The drying process, depending on the humidity of the environment, takes about 3 to 5 minutes. And similarly, the spray which we actually sprayed on the suspected fingerprint area also would have dried in 3 to 5 minutes. So now we are going to show you how to pick up the sample. We use what we call as a scraper. So we scrape this the area where we see the boundary of the Omni-Matrix. Can you hold just 1 minute? I just want to show that it's almost 100% of the sample is lifted, and it is collected on the scraper. This scraper, now we will deposit the collected sample with the Omni-Matrix into the collection tube. So now we are going to collect all the samples and then put it in the collection tube. Similarly, now we are going to collect the sprayed samples with the Omni-Matrix. This is the sprayed fingerprint suspected area on the wood surface. So we are collecting it with a spray like this, and it just comes up, and now we are going to deposit it directly into a tube, which is similar. In this case, we are using a tube where we could have used an Eppendorf tube also. In our next session, we will show you processing of these collected DNA samples. We just showed you the collection of the dried Omni-Matrix using a scraper. Actually, this scraper is an intermediate step. We have designed a better system for collection, and we are going to show you this is a special collection system which is connected to a tube, and we are going to, this is a beta tested using 3D printers, so we don't have, so this is a better collection system for the scraper or the dried Omni-Matrix, where the collected Omni-Matrix can be directly collected into the collection tube. After it is collected, as we know that it is all dried, and this is just a beta version, there will be more improvement to this. So we take away the scraper side, and then the sample is collected at the bottom of the tube. The sample is collected at the bottom of the tube, and this tube, after appropriate labeling, is then sent to the lab for DNA extraction and processing. This is session 3, and the sample in the collection tube in terms of swab or any of our Omni-Matrix have arrived in the lab. In the lab, they open the container, and normally with the swab, the front portion is cut off and inserted into a spin basket. So this is just the front portion of the swab which is cut off and inserted into spin basket with a tube. For our purposes today, we instead of using a spin tube, a microcentrifuge plastic tube, we are using glass tubes just for demonstration purposes. So we are going to insert it into a tube which contains the lysis buffer. The green, we have coded different tubes with green with different colors. So the green is the Omni-Mag lysis buffer manufactured by GeneLink, and in the other tubes, in the purple one is Promega DNA IQ system, DNA lysis buffer, prep filer from ABI, and then MTL buffer by Qiagen. Now we are going to transfer the samples which came in the Eppendorf tubes and into each one of these the glass tube. Normally in the lab they will not be transferring it, they will straight away add the lysis buffer into the microcentrifuge tube. The only reason we are transferring it today is for demonstration purposes to show you how the Omni-Matrix compares with the swab. With the swab, this is after like 2 to 3 minutes. In terms of in the swab, we are going to show you just pick up the tube itself, and you will see in the tube the color of the tube is the sample, the right tinge of the red color. The sample is being the blood, in this case blood, the biological fluid is being now dissolved into the lysis buffer. And if we are going to pull off the swab a little bit more, just to show you there is no way that 100% of the biological material will be released into the swab. The Omni-Matrix dissolves in the lysis buffer, and in this case it has been just like 3 minutes, 2 to 3 minutes, and you can see that there is hardly any Omni-Matrix left. What we will do is we will let it wait for about 5 to 10 minutes and then initiate and show you 100% dissolving. So the basis of the Omni-Matrix, it is a dissolvable sample collection device, and after adding to the lysis buffer it dissolves 100%. Thus there is 100% recovery of the sample if it was collected from the knife using any of the trace DNA samples, or from wood, or any other sample collection method. Whatever is collected is 100% soluble in the lysis buffer. It has been about 5 to 10 minutes that we allowed the Omni-Matrix and the swab to be soaked in the lysis buffer. In this case, what we did is we took the swab and actually centrifuged it also. Now we are going to take out the swab just to show you the color of the swab. The color of the swab will reflect, if you see, it has already been spun and there is solution at the bottom of the tube as well. But in terms of the swab, there's still the color. That color reflects sample retention, and that is what initially we're saying 20 to 80% of the sample is lost during this just absorption to the surface itself. Now we are going to show you the sample matrix, the Omni-Matrix, which were in each of these different lysis buffer. The color code is green, which was the Omni-Mag lysis buffer. The purple is the prep filer, and then from the ABI Thermo Fisher and the MQL. In all of them, I'm going to focus here. There's no matrix left, and all the matrix is totally dissolved and it is soluble. So whatever the sample was, 100% of the sample is dissolved in the in the lysis buffer, and then it can go follow through the procedures of automation extraction or manual extraction. The DNA collected is far superior and in almost 100% yield as from the Omni-Matrix as compared with the swab. Thank you so much.